Hello everyone. I've recently attended the Cerebellum Hackathon in Pavia, Italy. It was a 3-day workshop featuring a series of lectures and tutorials on cerebellum modeling and simulation. I was graciously invited to present our effort to simulate a realistic model of the cerebellum on SpiNNaker. I've been meaning to post more on this channel for a while now. I hope that this video is the first in a longer line of videos on neuromorphics in general and SpiNNaker in particular. Enjoy. To keep this on time, because maybe we finish all the presentations before lunch. So I'm gonna try and tell you about our current journey in trying to model the cerebellum on SpiNNaker. I'm gonna present a short overview of SpiNNaker and what it is, lessons we've learned from trying to model, or successfully modeling a microcircuit model on SpiNNaker, and then moving on to the cerebellum model that we're currently trying to run. So SpiNNaker is a project started in about 2005 with the express goal of putting 1 million ARM mobile phone processors in a very large computer. It was designed to be able to model about 1% of a human brain, or about 10 mice. The result of that project was a chip, also not confusingly called SpiNNaker, that contains 18 ARM cores that you can sort of see with the bluish arrow. That there's 18 processors with attached memory to each of them. And the most important bit probably is the router that I'm going to quickly cover here. So chips can communicate with each other. The whole platform is optimized for spiking neural networks. So we consider all spikes generated by neurons as being stereotypical. So the only thing we need to send to other cores or other neurons is where the spike came from, or address event representation. And because each chip is connected with 6 of its neighbors, we can send packets along through routers on each of the chips in hops. And we pride ourselves in having a multicast router technology, so a spike can be replicated on all links, all of the 6 links plus into the chip at once if needed. So a spike could go from chip 1.4 to chip 4.2, something like that, which is basically a point-to-point. You know where you're going, it's only one direction. But you can also have spikes that go to 3 chips, for example, or neurons on 3 different chips, to be more precise. And they can get replicated and sent very efficiently like this. This is the main, I guess, IP of SpiNNaker. So what do we have? We have these SpiNNaker chips. These are examples of 3D assemblies of chips. So you can see in the image that there's the chip that I showed earlier, plus a wire-bound SD-RAM, so large capacity but slower shared memory on top of the chip. We assemble these into boards of 48 of these beauties. And then you can assemble them into a 1,000,000-core machine that we have as of November last year, but the platform has been operational through the HPP Collaboratory since the 30th of March in 2016, when only half of the machine was built. I'm going to not go into a lot more detail on SpiNNaker, but I am going to have a shameless plug moment, because it's not every day you get your first book out. So this is going to be the SpiNNaker book, out in a couple of months, where you can read all about the thought process behind the design of the chip, how you create the chips, the machines, how you write software for something like this, and what kind of applications you can run on it, and all of that. It's going to be an open access book, but you can also buy it. So please buy it. Okay, on to the science. Sort of recently, there was a joint collaboration with Jülich Supercomputing Center to compare Markens, Diesmann's, and Potjans and Diesmann cortical microcircuit model running on Nest and running on SpiNNaker. And the 77,000 neurons and almost 300 million synapses, and we could show that on SpiNNaker you get very similar results to Nest. And this paper was published in Frontiers, although there was some interesting moment in time where some of our collaborators within HPP decided to do this model on GPUs, and they could show that it was faster. So Nest could do a 3 times slowdown. We could do a 20 times slowdown because SpiNNaker was not designed for, it wasn't originally designed for that kind of network. When my supervisor and the rest of the group were designing the chip, they were going off of, we're going to have 1,000 neurons per core with 1,000 synapses each, receiving firing of about 10 hertz, which is not realistic, we now know. But after a year and some redesign and rethink of the software, we were able to do it in real time at while also reducing the energy efficiency of the computation by an order of magnitude, so actually beating Nest, and the GPU, but not in energy efficiency though, at the cost of simply using more cores. But as I noted earlier, we have 1 million of these, so it's not a huge problem. It was barely felt for the big machine. This was about a 12-board job, and we have 600 of them. Now the challenge is to do the same thing, but for the scaffold model that was presented yesterday and probably will be presented again today in the tutorials. This model comes with very interesting features, but also challenges for us. It also has about 96,000 cells, all leaky, integrate, and fire, and about 4.2 million synapses, although they're organized differently than in the cortical microcircuit. I mean, you've heard these before, but it's a data-driven design of the network with connectivity generated based on experimental observation and constraints, with activity that matches experiments seen from PyNest and PyNeuron, both qualitatively and quantitatively, which is a problem for us. It would seem that SpiNNaker doesn't really like biology as much, but I'm sure we can make it work. Some of the features that we need to design for, as far as we're aware currently, is we need to account for the maximum number of spikes in a time step if we want to have this particular cerebellum model running in real time on SpiNNaker, which would mean individual Purkinje cells, for example, being able, or individual cores simulating the Purkinje cells, I should say, being able to deal with about 17,000 spikes in one time step. And I'm going to get to a few slides down to explain exactly how many spikes we could do for the cortical microcircuit that allowed us to do it in real time. We also have slight numerical constraints, as SpiNNaker does not have floating point units, and they're 32-bit ARM processors from 2005. So they're not particularly powerful. So currently the results that I'm showing on screen but not explaining are generated by slowing down the machine by about 2,000 times slower than real time. And they're not 100% correct. There's still more to do to both speed this up and increase the, let's say, the accuracy or the fidelity of the simulation. So what the lessons that we're trying to take from the cortical microcircuit are doing the software differently or making it much more specialized for the task at hand. So what are we currently doing with SpiNNaker? Well, SpiNNaker, again, has 18 ARM cores per each chip. Currently, if you go and run experiments, even right now, through the collaboratory, or I also have a board, physical board with me if the internet is bad here and if you want to try something. You will get on each processing core that is being used, you will get both software that updates the neuron state and updates the synapse state. If you want any Poisson input or anything like that, they would be, those kinds of inputs will be communicated via spikes. And even more, if you sometimes want delays that are not currently, so synaptic delays that are not currently supported by the tools, whatever that means, you will also get these kinds of delay cores that just act as buffers and resend spikes when needed. Well, this is the traditional mapping and how we've done things. And this is how the cerebellum model has been run currently, and how the microcircuit, the cortical microcircuit, has been run originally for the inefficient runs. Yeah. What we've learned from this is that we could do things differently by having dedicated cores that do either neuron updates or synapse processing, but not both. And in this scenario, synapse cores that only compute the synaptic inputs can communicate the current needed to the neuron through shared memory. So we're alleviating the problem of overloading the communication fabric. And this is exactly what allows us, well, this is one of the many reasons that allows us to run the cortical microcircuit in real time. What I wanted to say about this is that you see these synapse cores here. There are about 3 of them. It says SL, SU, and S, but I don't know how far from the back you can see this. They're basically split into 2 synapse cores per excitatory connections, and for the excitatory connections, and the red synapse core for the inhibitory connections. We can split this more finely. However, we've noticed that in order to maintain a real-time performance using this kind of architecture, each synapse core is able, in the time given, to process 20 spikes. 20 spikes for real-time performance for core. Which is not something we can do for 17,000 incoming spikes per one cell currently, which is the challenge that we need to face here, being able to use up to 800 cores just for synapse processing for a neuron if we're aiming for real time in the worst case scenarios. So this could be done. This is a potential solution. We're still investigating exactly if this is possible and how this can be done, but you could have your neuron somewhere on a chip, and then all the other chips around that one that's doing neuron state updates, all of them just doing synapses and communicating their updates once in a while at the end of a time step. Again, this hasn't been attempted ever on SpiNNaker, but if this is successful, then it would mean that any model with any sort of fan-in should be possible, allowing us to do all of the things that we promised with the SpiNNaker platform. Some of them being, you could have robot control systems using biophysically constrained models rather than simplified models of this, say, cerebellum. You can simulate the cerebellum like we have for the cortical microcircuit over days or weeks, at the cost of days or weeks, right? Because this would be real time. But it would also allow you to run online learning experiments that necessitate real-time interaction, say for robotics. If a robot is trying to learn something, you'd like it to do it now rather than in 2 days or 2 weeks. And also because we have full control over the platform, you could also induce pathologies and then study their behavior in, say, a robot or in a simulated environment like the NRP, the neurorobotics platform of the HPP. So in summary, we have been able to run cortical simulations on SpiNNaker in real time and with some tweaking of the software with an order of magnitude energy reduction per operation. Now for the cerebellum, it's a difficult problem that we're still analyzing. The key might be to scale off chips, so recruit nearby chips to do just synapse processing and nothing else. But this could open, if this is successful, this could open a huge avenue of further exploration. Thank you, and I'm on time.